In their most recent publication, Karen Labs utilized immunohistochemistry as an assay to visualize the presence of the GPRC5B G protein coupled receptor, among other receptors. This test helped to shine light on the importance of GPRC5B and its prevalence in the development of the lymphatic system. Immunohistochemistry is also important in a much wider sense throughout the medical field. Immunohistochemistry is used to diagnose many other diseases, such as an array of cancers. So, what exactly is immunohistochemistry? Well, simply defined, immunohistochemistry is a biological test used to show where proteins or antigens are present within a cell or group of tissues. As shown in this picture, these many proteins are visualized by the presence of the color where these proteins are. From this color presence, we would most likely be able to conclude that these proteins existed along the plasma membrane. Okay, so how does immunohistochemistry produce this color at the location of each protein? To perform immunohistochemistry, we first must collect a sample of our desired tissue and plate it onto a slide to put under a microscope. At this point, there will not be any fluorescent color present throughout the cells. Immunohistochemistry works largely with help from antibodies. It is commonly known that all cells have a widely different array of proteins within and outside the plasma membrane. Different sites on these proteins are recognized by specific antibodies, allowing the antibody to bind to the proteins. The first step in antibody binding takes place when the primary antibody, which is specific to one place on the protein of interest, binds to that protein. Then, a secondary antibody, which recognizes a specific site on the primary antibody, binds to the primary antibody. And this can be seen in the image below. The secondary antibody, which is now bound to the primary antibody, contains a fluorescent molecule. This fluorescent molecule can emit a light when in the presence of another molecule called a substrate. In this example, peroxidase or alkaline phosphate is the fluorescent molecule, and when this molecule interacts with the substrate, a navy blue color is produced. It is very interesting to note that the color emitted depends largely on the specific fluorescent molecule and substrate being used. All of these fluorescent molecules at work then combine to show all the desired proteins present, as we can see here. So this is an example of immunohistochemistry used by Karen Labs in their most recent publication. We can see the presence of 3 different colors of fluorescence in this test, indicating the search for 3 different types of proteins in their search to find the importance of many G protein coupled receptors in the development of the lymphatic system.